I'm very honored to take this talk. I would like to introduce myself a little bit. My name is Maura Carai, Senior Research Fellow at Tropical Feature Institute, James Cook University, Singapore. And today, I would like to talk with you about how tackle disease challenge in aquaculture using molecular and genomic tools. I'm going to give you a quick overview of the main topic I would like to go through it, beginning with aquaculture worldwide and in Singapore, followed by disease challenge in aquaculture, and how molecular and genomic diagnostic tool can address them. Then after, I also would like to introduce our new aqua health laboratory diagnostic, talk a bit about the environmental DNA surveillance, the current work project that we have done, and in the end, the conclusion, of course, some key takeaways. So, for starting, according to the FAO, Food and Aquaculture Organization, the global aquaculture production reached 130.9 million tons and in 2022, including aquatic animal and plants, of which 94.94 million tons were aquatic animal, represented by 40% on fin fish. The aquaculture market was valued $325.6 billion in 2025 and is projected to reach out a value of $418 billion by 2030. Just to give you some numbers about the importance of the aquaculture in the world. In Singapore, it's still quite important because aquaculture is not just business. It's a part of the national food security strategy. Singapore indeed imports over 90% of its food, including about 94%, 94% of the seafood. And therefore, aquaculture plays a strategic role in achieving the 30 by 30 goal. What is the 30 by 30 goal in Singapore? It is a national goal launched in 2019 by the Singapore Food Agency, commonly known as SFA, to produce 30% of the country's nutritional needs locally by the year 2030. To reach this point, SFA basically is focused on 5 main pillars, which of course including fish and aquaculture, eggs also, leaf and vegetable as the main source of protein and micronutrients. And then to achieve this goal and accomplish the task, also invest in agri-tech innovation, circular and sustainable system, and last but not least, regulatory and quality assurance in order to meet the national criteria. So in this direction, I want to also show you how is in this layout, the geographical layout of the Singapore. So in this slide, you can definitely see highlighted in blue. I don't know, do we have a point? This is the pointer. So highlighted in blue, this one, is the designer fish farming sites. So as you definitely can see, there is some geographical constraints. Singapore design fish farming zone both on land and sea, from high-tech recirculating aquaculture system to offshore cage. So the country is testing different models of sustainable intensification. Also, this slide shows some examples of the fish that are farmed in Singapore, such as Asian Seabass, red snapper, greasy grouper, and pompano, for instance. So, linking to the previous slides, Singapore aquaculture industry is facing multiple challenges, such as limited sea space, water quality, which of course help to the proliferation and the spreading of parasites, bacteria, and viruses, and therefore possible leading to possible outbreaks, which involve the loss of many stock of fish. Here, a list of examples of the pathogens that are challenging in aquaculture in Singapore. Scale drop disease, for instance, scale drop disease viruses responsible of scale drop syndrome in Asian Seabass, known also Barramundi or latest calcarifers, but not only Vibrio rvi, Streptococcus, Tenacibaculum maritimum, some example. What I wanted to underline that viruses, of course, can be responsible of outbreaks and the loss of many fish when the outbreaks occurs. But I think it would be nice just give you a little bit of thought in the sense that when an outbreak coming, why it happened? So let's think in this way. Fish, like human beings and any other animals, they have their own immune system. So technically they are they are structured to defeat possible pathogen in water, of course. But then when they are make sick, or I say weak, due to stress or poor quality of the water, poor nutrition, the fish, what happen? Immune system is not able to work properly, as happened to the human being when it's so stressed. And so the pathogen, they are there in the environment, make it easy to have access and make them sick. And this then cause what? Outbreaks. Why I want to say that? Because sometimes we are so busy to find the culprit or a possible outbreak, which is actually makes any sense. But also very important to understand what is the trigger of the outbreak. So try to do some step back and make a big picture of the situation where the fish was. Maybe if you implement the water quality, maybe if you make less stress the fish, he won't be able to get sick and won't be able to go through an outbreak. So moving forward, talking about what was the main topic of this lecture, molecular and genomic diagnostic tool applied to the aquaculture. So what we can say is definitely a very powerful, they are definitely powerful tools. The discovery of DNA double helix structured by Watson and Crick in 1954 definitely revolutionized the biology, setting the foundation for a molecular diagnostic. And over the decades, there have been developed different kind of molecular assay in order to improve this detection, detection in the sense of the pathogen, in order to improve the knowledge in this field, of course. Starting with PCR, for example, stands for polymerase chain reaction in 1980, followed by a quantitative PCR, then more recently digital PCR, and then after microarray, next generation sequencing, and lastly but not least, the CRISPR system, which is quite important because it's a very innovation technique recently adopted. And now I want to tell you about one of the quite powerful genomic tools that is being used in our university, Action Array, for instance. First of all, starting with describing some feature and how this is work, and then the application that we have done with that. So Axiom Array platform is a powerful, as I said, genotyping technology. Why so powerful? Because it's a high throughput screening of tens of thousands single nucleotide polymorphisms, SNPs, commonly named SNPs, across the genome. So you can think this system like a microchip. And every single part has 1,000 of probe, and each probe marks a different nucleotide. Okay. When you load your DNA in this, let's say, microchip, in this assay, what happens? The fragment will align and bind to the complementary segment that it can find in the chip. And that bond releases a signal according each nucleotide has a different probe, and each probe has a different mark is different color. So when this is happened, this bond ligation happen, the color will be emitted and detected by the software. And so in the end of the story, what we're gonna know? We're gonna know what is the variance of the nucleotide in our DNA that we have loaded in this system. And what are the applications? Because we have this beautiful system, but why is so important and why is so useful? No, using that we can do, as I mentioned, high throughput SNPs genotyping, nice, but also genomic selection breeding, GVAS, population genetic and diversity study, meaning we can monitor the genome of the population and better understand their diversity, which is still important in the sense of the breeding, especially when you're talking about fish. Parentage, assignment, and traceability, again, for the breeding. Copy number structure variant analysis. This is another thing important because a single mutation in a single nucleotide can affect the functionality of a gene. And these reflect in the expression of the gene and sometimes in a phenotypic way, reflecting a totally different phenotype. So having this kind of information can be definitely a useful tool for us for understanding the phenotype we are working with. So in this direction, the Tropical Feature Institute, under the Barramundi project, designed, in collaboration with Thermo Fisher, axiom array with 70,000 SNP, including 4,000 markers related to disease resistance, 29,000 linked to growth rate, an additional 80 candidate gene. What is the purpose, as I mentioned before? In order to making selection faster, more accurate, and more sustainable, and selecting the best individual with the genotyping on hundreds on individual, we can definitely pinpoint the best performer for the breeding. And now let's move to talking about the molecular tools. Polymerase chain reaction, commonly named PCR, which remains the backbone of molecular diagnostic, is the one I'm more confident with, actually. And the principle is very simple. Basically, allow the amplification of multiple copies of a specific DNA target. And this assay consists of 3 main steps: denaturation, annealing, and extension. I won't be boring you with the technical explanation, but what I want to underline the fact that when you have, using this technique, basically, repeating the cycle 30, 40 times, basically you amplify exponentially the amplification of your target. So in the end, if in your extracted DNA there are low copy of a specific target, let's say for example a pathogen, with a PCR you are able to detect this pathogen and visualize this pathogen, understand if it's there or not. However, as any other technique, there has also her limitation in terms of sensitivity. When the low copy of the pathogen is too low, a certain cutoff, basically is not able to be detected. Therefore, over the year has been developed additional technique, more sensitive, more accurate, giving more information, such as the qPCR, for example. I put this picture because it's quite familiar, I guess, unfortunately, for us, because this has happened, I guess, during the pandemic, when everyone on us, I think, has experienced the swab, the nasal swab, quite annoying and quite painful. However, what happened to this sample? Basically, they took the sample, they extracted DNA, and they used the DNA to test on a qPCR in order to detect if was or not COVID in there, and most importantly, if it wasn't there, how many copies of there was present in the specimen. Nowadays, we can also use another, let's say, upgraded version of the qPCR, we not offend the qPCR lovers, is called digital PCR. All these techniques use the basic same core of system, meaning the amplification, multiple amplification of the same target. But the results is different in the sense that conventional, traditional PCR, the first has been developed in terms of timeline, give you only an endpoint detection. So as a result, you know yes or no if your target, let's say a pathogen, is in your specimen or not, yes or no. The qPCR is more sensitive, and also it gives you the quantity of the pathogen that you have in your specimen, in copy per reaction, most likely. The digital PCR is another kind of qPCR in the sense that we don't have, the way it works is different in the sense that you have a sample that is being loaded and, let's say, partitioned in 1,000 of micro PCR. So for each sample, you have 1,000 of micro PCR at the same time. This is in practical what does mean? That increase the sensitivity of your assay. Because the qPCR, you have one sample and one test. In the digital PCR, you have one sample and 1,000 of PCR for that test. And that one, of course, gives you definitely a higher sensitivity, which is especially used when you have a sample with a low viral or pathogen copy. But also give you the quantity. So it's not endpoint detection. Also, they give you an absolute quantification of whatever you want to looking at in your specimen. And why I explain this technique? Because all this technique we are doing in our new diagnostic lab, Aquahealth, placed in James Cook University. Our scope is deliver precise, innovative, reliable solution, and most importantly, promote environmental stewardship, welfare, advanced aquaculture practice. In other words, we want to create a network of collaboration with the farmer, the industry, in order to increase our knowledge, help them to tackle the possible disease and the possible issue. In Aquahealth Laboratory, I'm working alongside with Professor Susan, and with her help, basically, we can offer a different range of services, from the molecular analysis for pathogen, for which I'm more expert, and Susan histopathological expert interpretation. Also, we do eDNA monitor and surveillance study. Why I'm mentioning eDNA? Because I want to speak about it. Environmental DNA, commonly named eDNA, is the new, let's say, new knowledge that right now is everyone try to work with. But what is it? So it's a very small fragment of genetic material, so DNA, that the organisms are left behind in their surrounding. Can be mucus, can be scale, can be skin cell, can be feces. And what happened to them? They're going to be accumulate in the soil. They can be accumulate in the water. And our purpose is collect them, analyze them. Why? Because in that case, we can not only study the biodiversity of the organism, in aquaculture particularly, we can detect the presence of possible pathogen before that pathogen is spreading, and it may cause outbreaks. And that's actually the aim of the pilot study I was running last year, for 6 months, based on environmental DNA study. So the main aim was to assess whether the results from flow cytometry, which is another technique I will talk later, and eDNA analysis can be an early warning tools to prevent possible disease outbreaks. So, as you know, I mentioned before, aquaculture is definitely vulnerable to infection disease. There are several pathogens that can affect the fish: scale drop, Seabaculum, Vibrio, Streptococcus. And this project was carried out in partnership with QIAGEN, leader in molecular diagnostic. Kitos is a startup driven microbiome analytics, and a Singapore aquaculture technology, a local farm, that allowed us to do the sampling. So I give you a little bit of explanation how we work during this pilot trial, this pilot surveillance project. The fish of interest for our study was Barramundi, latest calcarifer Asian Seabass. And we sampled there considering only 2 tanks, which I mentioned the name, but it's not that relevant. The most important thing is that the sampling occur over a 6-month period, 24 weeks, once a week. And this is the workflow that we did this kind of study. So basically, we collected 3 samples for the eDNA per tank per week, and 3 samples for the DNA, so basically 3 samples from fish, per tank per week. We extracted them, and then this DNA was assessed on digital PCR probe based for 3 different pathogens, specifically Vibrio RVI, Seabaculum maritimum, and Streptococcus inem. Kitos, instead, was in charge to running the flow cytometry analysis for Vibrio species and Seabaculum species. So the flow cytometry is less, let's say, specific than a digital. Digital can detect the species of the pathogen. Flow cytometers stop to the level of the genus. And for doing that, they collected 4 water samples per tank weekly. Here, the preliminary results I got for tank D3. On the left side, I have reported the copies per sample, okay, of the eDNA that's been tested for the 3 different pathogens: Strepto, Tena, and Vibrio. As you definitely can see, the the gray line showing the consistent presence of Vibrio RVI overall the period of the sampling. Quite high, always 1,000 copies per sample. Also, Seabaculum is quite present, but less consistent. Streptococcus instead appear time to time in some week. These results does not reflect the same results we have from the fish. So we tested the DNA from the fish to see if the fish was affected by this pathogen. And we didn't find the presence in the fish of Vibrio, as you can definitely observe, even Tena, just a spike here of Streptococcus between 4 and 5 weeks. These results, what is telling us? On the right side, I reported the weekly mortality rate that was registered over the period of the sampling, and also an average of the weight of the fish. As you definitely can see, there is some dead fish over that period with, let's say, a peak around week 9. So having not found any of these pathogens in the fish, meaning that basically those pathogens, they are not responsible for the death of this fish. So there is no correlation between the mortality registered and the pathogen that we have analyzed in tank D3 for that period. Same analysis we run on the tank D6. Again, in the water, we definitely found the Vibrio over the period of the sampling. Even Seabaculum and Streptococcus a little bit more consistent. In the tissue, we can definitely observe a presence on Strepto, again, week 4 and 5, and a little bit also in week 12 and later on, no Vibrio presence. And again, what this is meaning that these kind of pathogen, they are not the responsible of the mortality, instead registered and recorded during that period on the right side. Here, the mortality is a bit higher than before the other tank. Indeed, in week 10, the farmer sent the sample for analysis and find out they were positive for other pathogens, whereas they were still negative for Vibrio RVI even in week 15. So conclusion, those pathogens we have analyzed on digital PCR for this particular in this particular tank, they are not related with the mortality of that fish. And these are the results for the flow cytometry analysis. So the water sample analyzed in flow cytometry show up what? In Vibrio, they found a level of cell per ml reported on the y-axis. Vibrio species between 10,000 and 100 cells per ml. On the opposite, Tenacibaculus species wasn't detected at all over all the period of the surveillance. So basically, the presence of Vibrio here confirm the presence, of course, we're talking about Vibrio species, not Vibrio RVI. So Vibrio species included also Vibrio RVI, but also other species as well. So reflecting in a certain way the results that we found on digital. So our first set of conclusion regarding the surveillance project tell us that definitely we need to extend our analysis on digital and qPCR to other pathogens, because it would be interesting to have a panel of pathogen results and see if there is any correlation with the mortality. Again, as I said before, there are also other factors that affect the mortality of the fish. So we need also to consider the water quality. We need to consider the big picture around the fish. Moreover, it would be even much useful develop multiplex digital PCR assay. Why? Because having a multiplex reduce the number of the assay to run. I mean, increase the number of the pathogen you are able to detect in less time. And of course, in this way, cut also the cost, which is the most important things for the farmer. But the time is another important thing to keep in mind, especially when this has happened. So we need to be prompt. When a farmer contacts you and asks you for analyze some sample, we need to be quick, we need to be prompt, because outbreaking happen in few days, and in few days you can lose a lot of fish, even all the fish, if you're not quick enough. So it's good if we have in place proper and quick response in order to help them to address the problem properly. Moreover, during this surveillance project, we collected the water quality parameters, so it would be also nice try to combine them with the results from the molecular flow cytometer to find if it exists, of course, a patterns. Because if you find a pattern, a model that is recurrent, you basically understand what is trigger the possible outbreaks. Let's say the pH becoming too acid, or the oxygen dissolved too high. So some combination of this factor together that can maybe combined with the digital PCR that tell you something present, some pathogen in the water, they allow you to be quick in response and give you them the right solution to tackle the problem. And of course, another thing we would like to do is establish a national or regional aquaculture health surveillance program. In other words, involve more than one farm, because if we have more farms, we can extend our study, increase the number of results, and be able to better have a solid results to consider. This is for the surveillance. But for the conclusion of the lecture instead, I would like to tell that, of course, molecular genetic tools are super important and useful. However, we need to combine them, collaborating with the farmer. So we need to integrate them across the production chain matters. So there must be a two-way communication, because technology is nice and useful, but we need to tell them our system, and they need to communicate with us what they are doing in order to have a better picture of the situation and find the right solution. Also, again, early and accurate diagnosis saves stock and cost, and definitely molecular tools help in this way. Genomic information help for transform breeding programs, and all these kind of tools help to develop a better solution and reach the goal for the food security 3x3 of Singapore. Collaboration is essential and fundamental. It's a key aspect. We need to collaborate industry, advanced farmer, researcher, diagnostic lab, because information must be shared. We need to create a sort of network and a relation in between in order to have a proper plan to apply. In the end, I would like to thank of the people that be involved in this collaboration, in this surveillance project. James Cook University Singapore, Tropical Fish Institute, and of course Singapore Aquaculture Technology, the farm from where we have sample, QIAGEN, and Kitos. Thank you. Okay, perhaps let's start with some question within these people. Okay, yes.
Thank you. I have a question.
Yes.
Earlier, you mentioned COVID, coronavirus, the pandemic.
Yeah.
And during that time, everyone is familiar. We had the swab test, and you mentioned it as well in your slides. I remember that the swab test, they take 5, 10 minutes, 15 minutes, then you get the result. And it's very important. You mentioned surveillance. The World Health Organization used this to monitor how the pandemic was progressing. My question is, with AquaHealth Lab and its intention of doing this surveillance, is it the same thing that you could get the results very quickly? Is it 5 minutes, 15 minutes? Or another question is, do we have the same technology to be able to find out these pathogens in a farm, for example, and use very simple swabs, because everyone has access to this? Is it the case that farmers can have this ability to very quickly and on their own, without having you mentioned equipment like PCR. I don't think these are things you can buy from courts, for example. These are just my musings on where we are right now with these capabilities, this technology, and where are we going? Are we going towards maybe making this more accessible to farmers? Is this something worth pondering on, at least? Thank you.
Okay, that's a very good question. So it's something that every time I go to some convention, you can find a lot of people try to show up the new technology, the new system that we have in place, because it's actually a problem. As I said before, timing is the most important factor that we are struggling with, because when something's happened, we need to be prompt and we need to intervene quickly. That's why I was introducing the surveillance, because if we can play ahead, play in advance, we are able to defeat before this is happening. But when this is happening, I would say, unfortunately, there are systems in place that detect the presence or not. Let's say there was a convention I saw, there was this person that showed that you can take a sample of water and just drop something there. You can tell you if there is a Vibrio or not, which is nice. But sometimes we also need to be able to use the tools in the proper way. I want to say Vibrio, most of the time, it might be found in the water. So find out that you have Vibrio in the water, let's say Vibrio RVI, yes, okay, useful. But what does it mean? Do I need to treat my fish with treatment, or do I need to do something? Do I need to wait? because Vibrio is sometimes in there. What is important to know for Vibrio, for instance, is to know if reach out as a level of copy, number of in the water, that basically is a risk to develop an outbreak. So the technique, the YAT technique, the farmer try in every time to find the best technique to tackle the problem. And on the other side, we are here to try to help them to solve the issue, give our knowledge, our technology, our services, the experience. Let's say Susan, she's a veterinarian, she's quite experienced in aquaculture, so most of the time go there, try by side on the side to give expertise in order to tell them how to solve any possible issue. If something is in development, as you said, true, qPCR, digital PCR, instrument, equipment, costly, something that, of course, there is no point to having in a farm, especially because you must be trained to use for it. So we need something more practical. I think, again, as I said at the end of the presentation, it would be nice having this kind of channel of communication and collaboration between the farmer and who does this work like us, because we have the expertise to use these tools. They have the expertise to grow up the fish. We share information, we sharing this information, maybe we can develop a better plan to defeat the problem they're facing. Hi.
Sorry, no one else putting up my hand. Putting mine up.
Ah.
I put another question in the thing, but I'll just say it anyway. You put up the table showing different fish are susceptible to different diseases, and then you put you're wanting to breed the strong fish. But is that within a species, or can you crossbreed to improve resistance for some diseases? Like if one fish is resistant to a disease, can you do anything with other fish to get that resistance in them?
Wait, you're talking about, let me see if I understand your question. This one?
Well, you had a table and you said these are the diseases.
This one, okay, yeah.
And then different species are
correct.
susceptible.
Yeah.
But then if one fish has resistance to scale-grop disease, for instance, can you get some of that resistance from a different species and put it into those species?
Oh, well, it would be nice if you can do that one.
I don't know how close fish are.
Exactly. That's why I show you this one as a slide, because this is actually a sort of answer of your question. So we need to know what is the genome of our species. So what is the genomic configuration? And according to that, we are able to select which one is the most resistant, the easy to grow, let's say red snapper, the best color red. So that's why under this project, my colleague, because actually I've been part of this project, but not directly. I want to show up because it's a quite powerful technique and it's fair to talk about it. But definitely they work a lot for selecting the right treats, the right SNPs. There is a lot of work behind these handy things. And I'm still working now on the results and try to understand how we can deliver the results and use the results for improving the breeding.
Okay, so you're not getting into genetic modification through the Thank you. I think there were some questions in there.
Okay. I know that's how I read the question. I saw just the thing. Okay. What kind of collaboration work would you expect in the future? Well, definitely industry collaboration work. Let's say with Qiagen, we're still working with them, and I would like to extend this collaboration. And Ketos as well, so I want definitely reinforce this collaboration and look after other industry or partners in terms of technology would be definitely nice, because we are a laboratory, so we like to learn new technology, we like to improve our knowledge in technology that we can apply and help the farmer in solve the issue. And of course, collaboration with the farmer that we have in place. But that one is something that we are actually already doing pretty well. My colleague, they are doing pretty well, so it's something, but yeah, we can definitely extend collaboration. It would be nice, because another thing I would like to tell, our laboratory, diagnostic laboratory, is working on getting accreditation for the ISO 17025, which is quite important to give reliability in our results and open the chance to collaborate overseas. So it's nice if we can definitely collaborate overseas with other countries, so we have the chance to analyze possible specimen for our farmer in other places, Australia, Malaysia. Why not Italy? I'm Italian, I don't think so, maybe it's too far from them. So yes, definitely we are open to any sort of collaboration, and if you definitely want to contact me for this, I'm here and very free to talk about it. Yeah, another question. May I contact you sometimes by email? Yes, of course, you must too. We can talk about it, and if you're in Singapore, you can even pop in here and we can have a coffee. Any other question? No? And thank you so much for coming.